Hello viewers, welcome back to Anatomy Weekly. In our previous episodes, we tried to understand the anatomy of neurons. We also came to know about their types and the way they are connected to work with each other as well as to other effector organs to produce a required response. Continuing with the discussion, in this episode, we will try to understand some special nerve endings called receptors, and in what way the recent Nobel Prize in 2021 awarded to two eminent scientists, Davy Julius and Ardem Petapotian, is related to it. So friends, keep watching. The question, how we sense the physical world through somatic sensation, has fascinated humankind for millennia. During the first half of the 20th century, it became clear that temperature and pressure activate different types of nerves in the skin. The ability to sense and adapt to the environment is essential for survival in all organisms. In humans and other animals, somatic sensation arises from the body surface or internal organs and endow us with the sense of touch, proprioception, pain, and temperature. These are vital functions allowing organisms to continuously adapt to changes in the external as well as in the internal environment. An individual receives information from outside and from within the body by special sensory nerve endings called the receptors. The receptors act as transducers, that is, they receive mechanical stimulus and convert it into nerve impulses. The receptors are in fact sensory nerve endings specialized for reception of stimuli and transmitting them in the form of nerve impulses. These receptors can be classified in many ways, and one of the common classification is into functional types and anatomical types. The functional classification is based on the kind of information the receptors provide. They can be exteroceptors, where they provide information of touch, pain, temperature, and pressure. The proprioceptors provide information about the state of contraction of muscles, of joint movements, and positions. Interoceptors provide information from viscera and blood vessels, whereas the mechanoreceptors can sense mechanical deformation.
In a similar way, chemical and thermal stimuli are perceived by chemoreceptors and thermoreceptors. Nociceptors respond to any stimuli that bring about damage to the tissue, which is usually perceived as pain, discomfort, or irritation. There are photoreceptors stimulated by light, as for example the rods and cones of retina, and osmoreceptors which respond to changes in the osmotic pressure. Now, the classification based on anatomy or structure classify the receptors into two types: non-encapsulated and encapsulated. The non-encapsulated receptors do not show any particular specialization of the structure. They are in fact free nerve endings that are widely distributed in the body tissue, such as skin, cornea, periosteum, and can be either myelinated or non-myelinated. Most of these endings carry pain sensations, but they are also sensitive to temperature, touch, pressure, and tickle sensations. Other non-capsulated receptors like peritracheal root hair plexuses are stimulated by light touch, causing movement of hair present in the skin. Tactile discs of Merkel are expanded disc-like nerve endings in the germinative epidermal layer of hairless skin that transmit information about the degree of pressure exerted on the skin. On the other hand, the encapsulated receptors are enclosed in a capsule derived from the surrounding non-neuronal cells. For example, the tactile corpuscle of Messner found in the skin in those areas where tactile sensitivities are extremely well developed. The corpuscle is supplied by several myelinated nerve fibers, and they are responsible for two-point discrimination. The Pacinian corpuscle are the largest and the most numerous encapsulated receptors. They are rapidly adapting mechanoreceptors that are particularly sensitive to firm pressure. The end bulbs of various types, which consist of Multiple encapsulated branch nerve terminals are also found. Bulbous corpuscle are spherical and found mainly at the mucocutaneous junctions. Genital corpuscle are slightly different from bulbous corpuscle and occur in the genital skin. Similarly, raffinase corpuscle are some spindle-shaped structures located into the dermis of hairy skin. These are stretch receptors which respond when skin is stretched, causing stresses in dermal collagen. Apart from all these types of receptors, there are receptors present in muscle, tendon and joints. These receptors senses the movements occurring at the individual tissue level and helps us get the feedbacks and thus the desired movements.
Now let us come back and find out exactly what was the contribution from the two eminent scientists David Julius and Ardem Patapotian towards discovering the mechanism of temperature and pressure reception, for which they were awarded the Nobel Prize for Medicine or Physiology in October this year. Prior to the discoveries of David Julius and Ardem Patapotian, our understanding of how the nervous system senses and interprets our environment still contained a fundamental unsolved question. As discussed by now, we have understood that the receptors are transducers converting different inputs into electrical impulses. But how? That was the pertinent question until these two guys discovered it specifically for temperature and mechanical stimuli. In the latter part of the 1990s, David Julius saw the possibility for major advances by analyzing how the chemical compound capsaicin causes the burning sensation we feel when we come into contact with chili peppers. Capsaicin was already known to activate nerve cells, causing pain sensation, but how this chemical actually exerted this function was an unsolved riddle. After a laborious search, Julius and his co-workers found out a single gene that was able to make cells capsaicin sensitive. Once the gene for capsaicin sensing has been found, further experiments revealed that the identified gene encoded a novel ion channel protein, and this newly discovered capsaicin receptor was later named TRPV1. When Julius investigated the protein's ability to respond to heat, he realized that he had discovered a heat-sensing receptor that is activated at temperatures perceived as painful. The discovery of TRPV1 was a major breakthrough, leading the way to the unraveling of additional temperature-sensing receptors. Independent of one another, both David Julius and Ardem Patapotian used the chemical substance menthol to identify TRPM8, a receptor that was shown to be activated by cold.
While the mechanisms of temperature sensation were unfolding, it remained unclear how mechanical stimuli could be converted into our senses of touch and pressure. Researchers had previously found mechanical sensors in bacteria, but the mechanism underlying touch in vertebrates remained unknown. Ardem Patapotian, working at Scripps Research, wished to identify the elusive receptors that are activated by mechanical stimuli. Patapotian and his collaborators first identified a cell line that gave off a measurable electric signal when individual cells were poked with a micropipette. It was assumed that the receptor activated by mechanical force is an ion channel, and in a next step, 72 candidate genes encoding possible receptors were identified. These genes were inactivated one by one to discover the gene responsible for mechanosensitivity in the studied cells. After a tiring search, Patapotian and his co-workers succeeded in identifying a single gene whose slicing rendered the cell insensitive to poking with a micropipette. A new and entirely unknown mechanosensitive ion channel had been discovered and was given the name piezo-1 after the Greek word for pressure. Through its similarity to piezo-1, a second gene was discovered and named piezo-2. Sensory neurons were found to express high levels of piezo-2 and further studies firmly established that both piezo 1 and piezo 2 are ion channels that are directly activated by the exertion of pressure on the cell membranes. The breakthrough by Petapotin led to a series of papers from his and other groups demonstrating that the piezo 2 ion channel is essential for the sense of touch. Moreover, piezo 2 was shown to play a key role in the critically important sensing of body position and motion, which is known as proprioception. In future work, piezo 1 and piezo 2 channels have been shown to regulate additional important physiological processes including blood pressure, respiration, and urinary bladder control.
The groundbreaking discoveries of the TRP-V1, TRP-M8 and piezo channels by this year's Nobel laureates have allowed us to understand how heat, cold and mechanical force can initiate the nerve impulses that allow us to perceive and adapt to the world around us. The TRP channels are central for our ability to perceive temperature. The piezo 2 channels endows us with the sense of touch and the ability to feel the position and movement of our body parts. Intensive ongoing research originating from this year's Nobel Prize awarded discoveries focuses on elucidating their functions in a variety of physiological processes. This knowledge is being used to develop treatments for a wide range of disease conditions, including chronic pain. Folks, this is all we have for you today. Stay tuned to Anatomy Weekly. Keep watching, keep learning. We'll see you next time. Thank you.